Hello, and welcome to part 2 of our 3-part series on proteins. There they are. Here we go. So the next thing we talked about, kind of the basics of what proteins are made of, amino acids, how a polypeptide is formed by elongating the chain, using dehydration synthesis reactions and forming peptide bonds. So now we're going to talk about what it takes to make a functional protein. So a protein that can do one of the functions we talked about: enzymatic function, structural function, transport, regulatory, all that kind of stuff. So a functional protein consists of one or more polypeptides that is very, very precisely, and I cannot stress this enough, precisely twisted, folded, and coiled into a unique shape, okay, to give the unique structure so that it can have its proper function. So structure and function together again. So this is just kind of a picture of, this is a ribbon model. This shows you all the structures we're about to talk about right here. These are beta pleated sheets. TS. This right here is an alpha helix. So in a minute, when I talk about these things, that's what you can see, how kind of they're all folded up. And then if you have a space filling model, this is kind of the shape of the protein. It has lots of grooves and dents and things like that help it perform its function. And if it doesn't have that exact structure, it will not perform its function. Okay? So what determines the three-dimensional structure is the sequence of amino acids in the polypeptide. That determines the structure, and then, of course, the structure determines function. 4 levels of protein structure. So there are 4 levels of protein structure. Primary structure, like this, of a protein is the unique sequence of amino acids. Secondary structure consists of coils and folds in the polypeptide chain. Tertiary structure, interactions between the side chains, or those R groups that make each amino acid unique. And then quaternary structure, which is when a protein consists of multiple polypeptide chains. So let's start off with our primary structure, is just the straight up sequence of amino acids in a protein. So it's like the order of letters in a very, very, very long word. And primary structure is determined by genetic information. It's determined by your DNA. So let's look at a picture of primary structure. Again, this is kind of a zoom in of our amino acid chain. And you'll see there's kind of the main chain. We've got amino group, carbon, carboxyl group, amino group, carbon, carboxyl group, amino group, carbon, carboxyl group. And then we've got those R groups off to the side. Okay, so when we talk about kind of our main chain, we're talking about just the common parts of the amino acids and not the R groups. And our primary structure is just a sequence. We've got glycine, then proline, then threonine, then glycine, then threonine, then glycine, etc., and so on, all the way down to the end. Once we have our primary structure, once the protein is made, it starts to fold up, okay? And the second step, so primary structure is always first. The second step is secondary structure. Secondary structure results from our good old friend hydrogen bonds between the constituents of the polypeptide backbone. So just that main chain. It does not involve the R groups. There are 2 types of secondary structures. Remember that I just talked about. One is called an alpha helix, and the other one is called a beta pleated sheet. Here's what they look like. So you have interactions between the main chain here. If it happens to form a coil like this, okay, there's my hydrogen bonds. It's called an alpha helix. If it forms these kind of parallel sheets, that's a beta pleated sheet. And here's my hydrogen bonds again. There they are. Beta pleated sheets are kind of flatter, and alpha helices give the protein a more round shape. So once we have secondary structure, we can move on to our tertiary structure. Tertiary structure is determined when the R groups start to interact. So first the backbone interacts, then the R groups interact, okay? And because the R groups are what gives amino acids their personality, their flavor, their character, the interactions between different R groups depends on what those R groups are. So remember from the last screencast, part 1, there were hydrophobic R groups, there were hydrophilic R groups, there were charged R groups, there were polar R groups, there were nonpolar R groups. So the interactions involve those kind of properties. So the interactions can be hydrogen bonds, as always. They can even be ionic bonds. If you have charged R groups, they can form an ionic bond. You can have hydrophobic interactions between hydrophobic amino acids. Van der Waals interactions, there can be covalent bonds called disulfide bridges if there's a sulfhydryl group involved in the amino acid. And all of these things reinforce the higher order structure of the protein folding. So right here, what this ribbon represents is an alpha helix. And then you'll see, or the secondary structure of a protein, then you'll see there can be interaction with hydrogen bonds between these polar. Here's a hydrophobic interaction between nonpolar R groups. Here's an ionic bond between charged R groups. Okay, here's one of those disulfide bridges if there's some sulfur involved. Okay, and this helps the protein to fold up even further. So at this point, once you have tertiary structure, you have a complete folded polypeptide. But sometimes that's not enough for it to be functional. Sometimes you need to take 2 complete folded polypeptides and kind of stuff them together in order to form a functional protein. Oh, actually, before we go here, so here's kind of tertiary structure, and you can see there's these are all beta sheets. This is an alpha helix, and then they're all kind of folded up back on top of each other. So again, once you take something like this structure, this tertiary structure, and combine it with another protein that has tertiary structure, you can get what's called quaternary structure. Okay, and quaternary structure, 2 or more polypeptide chains that form one big protein. And these quaternary structure has subunits. And it can have 2 subunits, 3 subunits, 4 subunits. It usually doesn't get too much bigger than that. Collagen is an example of a protein that consists of quaternary structure, that has quaternary structure as functional protein. Hemoglobin, we're going to talk more about this in the concluding part of our protein series, is a globular protein, has 4 polypeptides, 2 alpha and 2 beta chains. And so let's look here. So here's an example of collagen, and you see it's got 3. So it's got the orange, the green, and the blue. There are 3 individual polypeptides with tertiary structure, and then they interact to form kind of this rope structure, and then has quaternary structure. So this one would have 1, 2, 3 subunits. Okay, and here it is all kind of on one page, mostly. So you'll have your primary structure, which is the amino acid sequence, then secondary structure, which is alpha helices and beta pleated sheets, interactions in the backbone. Tertiary structure starts to bring in the R groups, and you get a folded up protein. And then so a folded up polypeptide, if one polypeptide isn't enough, can interact with, I think there's 4 of them here. Yeah, 1, 2, 3, 4. That gives us quaternary structure. And you finally have the proper structure so you can have the proper function on your protein. Okay, so, and there are very strict rules about folding up. You have to start with primary, go to secondary, go to tertiary, and then go to quaternary. And when you unfold it, it's the reverse. Quaternary unfolds into tertiary, tertiary unfolds into secondary, secondary unfolds into primary. You can't like skip a step. Okay, so this folds up, and this unfolds. And this unfolding has a special name. It's called denaturation. We're going to talk more about denaturation later on. Okay, I think that's it. End part 2.